Hello, everyone. Welcome back to Meds.Concepts. Today's lecture on transcription in prokaryotes. In the previous lecture, we discussed the basic concepts of transcription. Today, we will focus specifically on bacteria, initiation, elongation, and termination mechanisms. We will also discuss bacterial RNA polymerase, sigma factors, promoter recognition, nose proteins, intrinsic and rho-dependent termination, and transcription inhibitors. Prokaryotic transcription. Prokaryotic transcription occurs in the cytoplasm because bacteria do not possess a nuclear membrane. An important feature of prokaryotes is that transcription and translation are coupled processes. As RNA is synthesized, ribosomes can immediately begin translation. Prokaryotes contain a single RNA polymerase responsible for synthesis of all RNA types. Transcription occurs in three major stages: initiation, elongation, and termination. Many bacterial mRNAs are polycistronic, meaning a single mRNA molecule can encode multiple proteins. Bacterial RNA polymerase. The bacterial RNA polymerase consists of multiple subunits. The core enzyme contains alpha-2, beta, beta-prime, and omega subunits. This core enzyme is represented as alpha-beta-beta-omega. When sigma factor associates with the core enzyme, the Holoenzyme is formed. The Holoenzyme is represented as alpha-beta-beta-omega-sigma. Each subunit has a specific function. The alpha subunits help enzyme assembly and promoter interaction. The beta subunit performs catalytic RNA synthesis. The beta-prime subunit binds DNA. The omega subunit stabilizes the enzyme. The sigma factor provides promoter specificity and helps transcription initiation. After transcription initiation, sigma factor is usually released and the core enzyme continues elongation. Sigma factors. Sigma factors are specialized bacterial proteins that guide RNA polymerase to specific promoters. Different sigma factors allow bacteria to rapidly adapt to environmental changes. Sigma 70 is the housekeeping sigma factor responsible for expression of essential genes during normal growth. Sigma 32 controls heat shock response genes. Sigma 54 regulates nitrogen metabolism genes. Some bacteria also contain anti-sigma factors which inhibit sigma factor activity under specific conditions. An important concept is that sigma factors provide promoter recognition specificity. Promoter structure. Promoters are specific DNA sequences where RNA polymerase binds to initiate transcription. In bacteria, two important promoter consensus sequences are present. The first is the -35 region with consensus sequence, TTGACA. The second is the -10 region, also called the Pribnow Box, with consensus sequence, TATAAT. The transcription start site is designated as +1. Proper spacing between the -35 and -10 regions is extremely important for efficient transcription. The at-rich -10 region facilitates local DNA melting during initiation. Initiation of transcription. The RNA polymerase Holoenzyme specifically locates and binds to the promoter region. At this stage, the DNA remains completely double-stranded, forming what we call the closed complex, as shown in the top illustration. Then enzyme melts open approximately 17 base pairs of DNA around the transcription start site. This localized unwinding creates the transcription bubble, establishing the open complex where the template strand becomes accessible. The enzyme catalyzes the first phosphodiester bond formation. This marks the actual start of RNA synthesis, using incoming NTPs to build the very beginning of the transcript. The enzyme must clear the promoter. The structural tension causes the sigma factor to be released or significantly loosened, freeing the core enzyme from its initial anchor points. As the sigma factor falls away, a stable nascent RNA transcript of about 12 to 15 nucleotides is successfully generated. In processive elongation, the stable RNA-DNA ternary complex is now fully locked in. Abortive initiation and promoter escape. During early transcription, RNA polymerase often produces very short RNA molecules called abortive transcripts. These transcripts are usually less than 10 nucleotides long. At this stage, RNA polymerase repeatedly moves forward and backward on the promoter without leaving it. This process is called abortive initiation. During this period, downstream DNA accumulates within RNA polymerase in a process called DNA scrunching. The energy stored through scrunching helps RNA polymerase break promoter interactions. Once RNA reaches approximately 10 nucleotides in length, promoter escape occurs. Sigma factor region 3.2 is displaced and RNA polymerase enters productive elongation. Elongation. During elongation, the core RNA polymerase synthesizes RNA continuously in the 5' to 3' direction. The transcription bubble moves along DNA while maintaining a short RNA-DNA hybrid region. The elongation rate in bacteria is approximately 40 nucleotides per second. As RNA polymerase moves, positive supercoils accumulate ahead of the enzyme and negative supercoils form behind it. DNA gyrase removes positive supercoils, whereas topoisomerase I relaxes negative supercoils. Certain drugs such as cordycepin can inhibit elongation by terminating RNA synthesis. Role of Nus proteins. Nus proteins are transcription accessory proteins that regulate elongation and termination. Nus A binds RNA polymerase and promotes transcriptional pausing. It also stimulates intrinsic termination and interacts with nascent RNA. Nus B specifically recognizes and binds to the box A RNA element. Forms a heterodimer with Nus E to mediate processive antitermination. Nus E, S10, identical to the ribosomal protein S10. Binds to Nus B and links the transcription complex to the ribosome. Nus G enhances elongation processivity and plays an important role in rho-dependent termination. Nus G can also couple transcription with translation by interacting with ribosomes. Thus, Nus proteins fine-tune transcription efficiency and termination mechanisms. Intrinsic, rho-independent, termination. Intrinsic termination, also called rho-independent termination, does not require any additional protein factor. The terminator sequence contains GC-rich inverted repeats, followed by a U-rich region. During transcription, the inverted repeats form a stable hairpin structure in RNA. Formation of the hairpin causes RNA polymerase to pause. The weak UA base pairs in the RNA-DNA hybrid then dissociate easily. As a result, RNA is released and transcription terminates. This mechanism depends mainly on RNA secondary structure formation. Rho-dependent termination. Rho-dependent termination requires the Rho protein. Rho is an ATP-dependent RNA helicase and exists as a homohexamer. Rho binds a specific RNA sequence called the RUT site, which is generally rich in cytosine and poor in guanine. After binding RNA, Rho moves in the 5' to 3' direction using ATP hydrolysis. Rho catches up with post-RNA polymerase and disrupts the RNA-DNA hybrid. This releases the RNA transcript and terminates transcription. Nus A assists rho-dependent termination by enhancing polymerase pausing. Transcription-translation coupling. One unique feature of prokaryotes is coupling between transcription and translation. Because bacteria lack a nuclear membrane, ribosomes can begin translating mRNA while transcription is still occurring. This allows extremely rapid gene expression. NUSG plays an important role in coupling transcription with translation by connecting RNA polymerase and ribosomal proteins. Coupling also helps coordinate gene regulation and increases cellular efficiency. Transcription inhibitors. Several antibiotics and chemicals inhibit transcription. Rifampicin binds the beta subunit of bacterial RNA polymerase. It blocks transcription initiation by preventing early RNA chain formation. Mutations in the beta subunit can confer rifampicin resistance. Actinomycin D intercalates into DNA and inhibits elongation. Cordycepin lacks a 3' hydroxyl group and therefore terminates RNA chain elongation. These inhibitors are important both clinically and experimentally. Let us revise some highly important concepts. Sigma factor is required for promoter recognition and initiation. Sigma factor is released after promoter escape. The core enzyme performs elongation. The -10 region is called the Pribnow Box. Sigma 32 regulates heat shock genes. Intrinsic termination requires hairpin formation. Rho is an ATP-dependent helicase. Rifampicin targets the beta subunit. NUS proteins regulate elongation and termination. These concepts are frequently asked in competitive examinations. Thank you. If you found this video helpful, please like, subscribe and share it with your friends.